Welcome to the Molecular Therapy Podcast. I'm Dr. Rory Bricker-Anthony, Scientific Editor of the Molecular Therapy family of journals. This episode features a conversation between the Editor-in-Chief of Molecular Therapy, Dr. Joseph Glorioso, and Dr. Kathy Steece-Collier from Michigan State University, Dr. Fredric Manfredsson from the Barrow Neurological Institute, and Dr. Jeffrey Kordower from Arizona State University. They will discuss an article recently published in Molecular Therapy by Steece-Collier and colleagues titled Disease-Modifying Multidimensional Efficacy of Putaminal CAV 1.3 shRNA Gene Therapy in Aged Parkinsonism Male and Female Macaques. In the new year, let's look forward to exciting new breakthroughs presented at the ASGCT annual meeting, taking place May 11th through the 15th in Boston. Start your new year by preparing your abstract for the world's largest gathering of cell and gene therapy professionals. Don't miss your chance to publish your abstract in Molecular Therapy and present your research at the annual meeting. The submission deadline is January 30th. Visit annualmeeting.asgct.org to submit your abstract and register today. And now, Drs. Glorioso, Steece-Collier, Kordower, and Manfredsson.
Okay, thank you so much, Kathy, Jeffrey, and Fredric for joining us today to talk about some of the work that you've done in monkey models of Parkinson's disease. And it's a very important area, obviously. And so I think that we've created a number of questions that hopefully, can get our audience up to speed for those who are not expert in this disease. And the first question I had was, could you give us a brief history of Parkinson's disease, the age of onset, the size of the patient population in the U.S. and elsewhere? And is this population expected to grow?
So Parkinson's disease came about in 1917 when James Parkinson wrote his monograph essay on shaking palsy. But he didn't examine patients. He actually was not really a physician. But he saw subjects in the streets of London, and he saw some of them who had tremor and they had slowness of movement, and he wrote about it in this book, and that's how Parkinson's disease got coined. Age of onset is kind of different in different places, but normally it's about 62, 63 years old, but it can occur much earlier. The much earlier cases tend to be genetic forms of Parkinson's disease. I don't think I'm telling tales out of tune here, but because it's public knowledge, but Michael J. Fox got Parkinson's disease when he was 29 years old. That's really on the early side. In terms of patient population, in 2015, it was estimated that 6.5 million people around the world had Parkinson's disease, probably 1 to 2 million in the United States. But it's estimated by 2040 there were going to be 14 million or 15 million people around the world with Parkinson's disease. The reason for this big uptick, and in fact, Parkinson's disease is the fastest growing neurodegenerative disease. It's even faster than Alzheimer's disease. But there are a couple of reasons of why there's so much more Parkinson's now. And one of them is pesticide use. And pesticides are known to cause a form of Parkinson's disease. And the other one that's kind of interesting to me is that people don't smoke cigarettes anymore. And smoking cigarettes is protective against Parkinson's disease. So that, now, I always say this when I teach class, I go, don't go home to your parents and tell them Dr. Kordower said you should be smoking cigarettes so you don't get Parkinson's disease. But that's why this incredible uptick in the number of patients is taking place. With regards to pesticides, there was just an interesting paper that came out. If you look, are you a golfer, Joe?
No, I'm not. I'm a tennis
Okay,
player. At least I
that's
used to be.
that's a good thing because if you live between 1 to 3 miles of a golf course, you have a dramatic increase of chance of getting Parkinson's disease.
Yeah, I've
And,
held that information from my sister, whose backyard is a major golf course at a country club.
What's the mechanism behind that?
well, it's pesticide. They use
Oh,
a
it's
tremendous amount
pesticide to keep the greens.
yeah,
Yep.
Okay.
keep everything so clean.
Yeah, when I was a kid, they were still using DDT, right? So thanks so much for that introduction to the disease process. I mean, it's really an amazing development in the population. And I guess Parkinson starts probably much earlier than symptoms occur.
Mhm.
And
Undoubtedly,
yeah,
undoubtedly.
and so anyway, so maybe one of you can talk about the brain and how it's affected and what are the common symptoms, and maybe a little bit about the complicated genetics of Parkinson's disease and how this might affect therapeutic development.
Okay, I'll start off by talking about the brain regions affected and then the common symptoms. So Parkinson's is primarily involves a grouping of nuclei that are found in the brain, deep in the brain. It's a grouping called the basal ganglia. And so these nuclei are crucial for controlling movement, acting kind of as gatekeepers that promote desired movements and suppress unwanted movements. So if there's an imbalance in the basal ganglia nuclei, you can either have too much movement, like in Huntington's disease, or too few movements or difficulty with movement, like in Parkinson's disease. So in Parkinson's disease, the primary deficit within the basal ganglia is the death of dopamine-producing neurons in a nuclei called the substantia nigra. These substantia nigra dopamine-producing neurons send their projections up to an area called the striatum, which is another basal ganglia nuclei that is our target area, primary target area, because we're also interested in substantia nigra, but we may get into that a little bit later. But the loss of dopamine in the striatum, as those dopamine neurons are dying off in Parkinson's disease, is what results in the motor symptoms of Parkinson's disease. And these include tremor, rigidity or stiffness, and then bradykinesia or difficulty and slowness of movement. There are other areas of the brain that are affected, and these are thought to underlie the plethora of non-motor symptoms that for a long time were kind of ignored, but now are really at the center of some of the treatments and research that's going on right now. But some of these non-motor symptoms include digestive issues. Constipation is a major non-motor effect in Parkinson patients. Difficulty with blood pressure regulation, apathy or lack of motivation, and then cognitive decline. So,
Is that cognitive decline similar to what occurs in Alzheimer patients? Do you get degeneration of cells in the brain broadly, or?
yeah, it's thought that the basal ganglia itself is a cognitive motor aspect. I mean, there's both there's a limbic aspect, there's a motor aspect of this constellation of nuclei. So losing proper connectivity and function of the basal ganglia in and of itself can have a cognitive impact, but frontal cortex is also impacted. I think it's frontal cortex, is that right?
Yeah.
Jeff, Fredric? And that can definitely impact, cognition in patients. So
I see. So what about the genetics of Parkinson's? It's a very complex disease, and multiple genes have been appropriated as being important. I guess alpha-synuclein is probably the key one, right? And you ends up with Lewy bodies, tangles in the brain. And are these responsible for destruction of the termini of these dopaminergic neurons? Is that how that works?
yeah, Fredric, do you want to take that question? I think.
Yeah, sure. Yeah. So I mean, obviously, we're learning more and more on the genetics and the direct causes, and there's a lot of genetic risk factors. identified as well. But I think the truth of the matter, it gets to be really complicated because we're learning a lot of the genetics, but we're having a hard time dissecting what about this genetics is actually causing disease. I mean, some are simple. You have loss of function, for instance, GBA variants of PD, where you have loss of function. But in terms of proteins like, for instance, alpha-synuclein, yes, it's part of familial forms. The protein is also part of sporadic disease. Obviously, it's part of the Lewy pathology, Lewy neurites. But I think the field at large cannot agree or cannot decide as to why synuclein and why synuclein pathology causes disease. It's one of these really difficult proteins to study. It kind of shows up wherever in the cell you look. There seems to be synuclein associated with that organelle. You even have synuclein in the periphery and so on and so forth. So I think still, although we're learning more about the genetics themselves, they're not really helping us guide therapeutics. I think just coming from a gene therapy standpoint, I think a majority of the therapeutics being brought out is really more kind of pleiotropic in nature, in that, for instance, GDNF growth factors that's just aimed to provide a pro-survival environment, regardless of what the causes of disease are. But certainly, I mean, we're learning more and more, and at some point, hopefully, we'll get a much better understanding of these different forms of disease.
I think one of the other features, if I just jump in here for a minute, is that Parkinson's disease, like almost all neurodegenerative diseases, are co-pathology diseases. So 100% of PD patients over the age of 60 have a tauopathy. And about 30 to 40% of patients over 60 years of age have 4 different pathologies, stroke being a major one, mini strokes, and these TDP43 tau and alpha-synuclein. So it's not just the one alpha-synuclein gene that is causing the progression of Parkinson's disease.
Yeah, I think that's one of the biggest changes in thinking in neurodegeneration over the last year or two.
So levodopa was used many years ago, still is, but it's not effective long term, I guess, and it's more of a treatment. It's not a cure. So what are the limitations of using levodopa to treat Parkinson's?
So Parkinson's disease, unlike a lot of neurodegenerative diseases, actually have many therapies that are available to individuals that are afflicted with this disease. However, even though we have levodopa, which is still, I believe, considered, a miracle drug, it certainly was in its heyday once dosing and those type of things were worked out. But there remain many unmet needs, even though there's a lot of different therapies that are available. And key most of these is the fact that we have no disease-modifying therapy at this point to stop the progressive death of those nigroid dopamine neurons. I would say a lion's share of the research is going on in the field to try and figure out how to stop that and what causes that cell death, like Fredric just talked about. But the other thing is that, again, we have all of these different, wonderful chemotherapies, which are like levodopa and dopamine agonists. But even though levodopa, which is the gold standard drug, even though it provides great benefit to many patients, it provides incomplete efficacy. And in many patients, it doesn't really work. And with disease progression, the efficacy wanes. And by incomplete efficacy, what I mean is that levodopa and dopamine agonists are not good at treating things like apathy, lack of motivation, which is very common in Parkinson's disease. And things like postural instability can actually be made worse by levodopa. So there's incomplete symptom benefit, even in patients that have great motor benefit from things like levodopa. And the other thing is, ever since the introduction of levodopa back in the, late 1960s, it was written about in that day and age when they first introduced it, that it's an interesting intellectual endeavor to give patients levodopa, but it will never be used in patients with Parkinson's because it has so many side effects. Patients were developing levodopa-induced dyskinesias very rapidly, and again, they had to give such high doses because they didn't know about giving peripheral decarboxylase inhibitors to prevent the conversion of levodopa to dopamine in the periphery. So it's been more than 60 years that we've known about levodopa-induced dyskinesias, and there's one FDA-approved drug that's partially effective in the subpopulation of patients for a short period of time, and it's contraindicated in about 25% of patients. So that's a real unmet need.
Yeah, it's a dramatic unmet need.
Yeah.
What is dyskinesia that's caused by?
Thank you. Yes, I meant to describe that. Dyskinesias are abnormal involuntary motor behaviors. So they can be chorea, which is, flailing, dancing-like activity, dystonias, abnormal, muscle tone. And they can be anywhere from just, annoying, excessive movements to being quite debilitating. There are patients that are so dyskinetic that, one person that I know here in Grand Rapids has to use walking sticks when he has his peak dyskinesias because he's so dyskinetic. Parkinson's is a disease that affects balance. And if you imagine yourself trying to walk on a balance beam and dancing, it really affects your ability to not fall. So falls are a big morbidity associated with, severe dyskinesias. And, we know of one celebrity that has had quite a bit of that, and people become wheelchair bound and need caregiver support. So
going a little bit further along into this, what are the best targets for gene therapy for Parkinson's, and of course, including your calcium channel gene?
I can jump in on this, but I think, so certainly, as I mentioned earlier, there are familial forms which are described as a loss of function. So GCase, GBA1, for instance, and there are active efforts in restoring function of that protein. Other trials have been neuromodulatory, trying to affect the electric output of various anatomical circuits within the brain. So essentially kind of a genetic therapy version of deep brain stimulation has been done. The other ones have been neurotrophic factors, GDNF and nurturin. Jeff was involved in the nurturin trials, had a very robust body of preclinical data. But up until now, there's been no clinical efficacy as the trials have been performed. We mentioned synuclein. Synuclein initially was thought to be a gain of function target, and there was a lot of preclinical efforts going after that protein, essentially as eliminating it. But over the last decade or so, the field has moved away from just simply eliminating, recognizing that it's probably an important protein, and now focusing more on perhaps approaches that disrupt its aggregation. Our therapy, obviously, normalizing these calcium channels, as we point out in our article, I think we're providing a multimodal benefit. So in one sense, as we deliver it to the striatal neurons, the postsynaptic component of this circuit, I think is neuromodulatory. If we get it into nigral neurons that have a high degree of calcium stress, I think it might be neuroprotective or neurorestorative. So certainly that holds great potential as well. But Parkinson's disease in general has been a popular target for gene therapy. And I think it has to do with the fact that you can do a targeted surgery with these injections, getting it into the circuit that's involved.
Maybe you could describe in more detail the role of CAV 1.3 in the, I guess it acts in the downstream of the striatum, downstream of dopaminergic neurons. Why is that such an important target, and what's the function of that channel?
So this will be my most lengthy dialogue, probably. But I will try and keep it succinct while also doing justice to its important role in our therapy. So CAV 1.3 calcium channels have been of intense interest as therapeutics for Parkinson's disease based on their localization in two areas in the basal ganglia. So one area where they're centrally involved is in the substantia nigra dopamine neurons. So in those dopamine neurons, CAV 1.3 plays a essential role in the pacemaking activity of those neurons. So those cells are tonically active, and the pacemaking activity is mediated in large part by the Cav1.3 calcium channels. So there's chronic tonic influx of calcium into neurons that have a very high metabolic load. And this results in high cellular stress with an influx of calcium and, again, the high metabolic load. And this is thought to underlie their vulnerability to neurodegeneration in Parkinson's disease. So it's thought to be very central to the death of those neurons. So the thought is, and there has been a clinical trial, Steady PD, where they tried silencing those calcium channels in the substantia nigra for neuroprotection. Unfortunately, it failed, and we can talk about that more later. But the target that we started targeting, and as Fredric alluded to a little bit, I think we're involving a little bit of the nigral Cav1.3 channels through some retrograde transport of our vector. But we're targeting Cav1.3 in the striatum. And in the striatum, Cav1.3 is localized to what are called spiny projection neurons. And they're called spiny neurons because the dendrites, which are the antennae of a neuron, of the spiny projection neurons, are studded with thousands and thousands of dendritic spines. And it's those spines that receive the input from the massive cortical input and other motor areas that are bringing information into the striatum. So these Cav1.3 channels are localized on the dendritic spines, and this positioning allows them to regulate the synaptic function of the inputting information from other brain areas. So they function as synaptic function regulators, and they regulate the plasticity of striatal neurons and their synaptic connections. So in Parkinson's disease, there's a decrease in dopamine in the striatum because the nigral neurons that project up there are dying off. Normally, dopamine modulates or acts as a brake for Cav channel activity. So in Parkinson's disease, when there's a decrease in striatal dopamine, these channels become dysfunctional or overactive. This results in rapid and profound changes in the structure of those spiny projection neurons. Largely, they lose a lot of their dendritic spines and ability to receive proper input. So there's these circuit changes that gives rise to miswiring that we've seen associated with levodopa-induced dyskinesias. So this complex and beautiful story led us to hypothesize that we know we can't prevent the decrease in dopamine, but can we impact the aberrant Cav channels by silencing them or decreasing their population in the medium spiny neurons? And if we could do that, could we ameliorate that striatal pathology that's associated with therapy dysfunction, specifically the dyskinesias? So that's the rationale for our target.
So I guess the dysregulation that comes from this overactive calcium channel is by using an shRNA, which I guess associates with the messenger RNA, and you have a risk degradation into an siRNA, and then reduction of the making of the protein, I think, if this is my understanding is correct, then you have less activation of neurotransmitters. What was remarkable is that in your study, you showed in these MSTP primate models that you could actually reverse the effects of levodopa causing dyskinesia. But in addition, it appeared that shRNA alone against that calcium channel also relieved Parkinson's, symptoms without even using levodopa. What do you think about that? I mean, that was a pretty remarkable finding.
It was really remarkable, that it occurred in such aged subjects and subjects that were, so Parkinsonian. I was very surprised that occurred. How it occurred, I will start by answering something, and then maybe, Fredric and Jeff can add in if they want. But so we believe that when we give our shRNA up in the striatum, we're correcting some of those miswired circuits. So we're restoring the morphology of the medium spiny neurons. We've shown that to some degree. but we haven't shown it necessarily as confirmation in this non-human primate model, but we believe that is occurring. But the other thing that we're noticing is, and I mentioned it earlier, we have evidence that there's retrograde transport of our vector in neurons that have not been killed off by the MPTP. So our model, on one side, we're actually killing off the nigral dopamine neurons, and Jeff, correct me if I'm wrong on any of this. So once we give intercarotid MPTP on that side of the brain, those nigral dopamine neurons are killed off because it's a very robust toxic insult. But then, in order to be able to have the animals self-care, we don't want to kill off both sides of the substantia nigra. So we give systemic MPTP. And what happens there is that some of the neurons exposed to this, systemic MPTP gets into the brain. It will kill some of those neurons, but it also just injures some of those neurons. So they lose their dopamine phenotype, they lose DAT, dopamine transporter. So there's this injury phenotype. They're not dead, but they're probably not functional. This occurs in Parkinson's disease. There's that same injury phenotype. It occurs in alpha-synuclein overexpression models. So we think that this is a really great model to emulate what might be able to happen in a patient. So those neurons are apparently, again, we see a decrease in the TH up in the striatum. So they've turned off their TH, but apparently they're still alive enough, I'll call it, to be able to pick up the vector, retrogradely transport it back to the nigra, silence those calcium channels, restore physiological balance, and we see a restoration of TH phenotype. And I'm not at liberty to say what else yet, but we see another marker that's phenomenally brought back to baseline that's involved with dopamine production. So it seems like we're restoring the function of some of the injured dopamine neurons, as well as impacting the integrity of the host. So, I think with the GDNF trials, again, correct me if I'm wrong here, Jeff and Fredric, but there's been really robust restoration of the TH fibers, sprouting of the TH fibers, but no functional benefit. My belief is that that's because there's pathology in the striatum. And, you can transplant new dopamine neurons or regrow old, or new dopamine terminals through trophic factors and stuff. If they have nothing proper to hook up with, or the circuitry of what they're hooking up with is abnormal, you're not going to get functional benefit. So I think our CAV therapy is impacting the circuitry aspect up in the striatum, and it's restoring the dopamine neurons. That's hypothesis, or speculation, but it's based on data, our data. So,
what do you know anything about the durability of this treatment? I mean, I guess it just depends on how long you get expression of the shRNA, right? Is this long term, or what's your sense of that?
Yeah, I guess I can jump in. Yes, I mean, just based on everything we know about AAV biology, right? And these are post-mitotic cells. One would expect that this would be active for the lifetime of the a neuron. And we're using these ubiquitous promoters that shouldn't get methylated. And as we're moving into also microRNA trials, we're using standard Pol2 promoters that we know they don't get methylated. So they should be active for the lifetime of that cell. And as a matter of fact, Jeff has published a couple of papers now on the nurturing trials postmortem, and I think looking out to 10, 12 years, and the activity of the virus is as expected. So this should be one and done, is the short answer to that.
Right. So you don't anticipate, redosing or anything like that? You don't think it'll be necessary?
No.
It could be deleterious if you redose, because if you use the same serotype twice, you're going to get an inflammatory response.
Yes, that's right. What role do mitochondria play in this, in the MPTP model?
Yeah, so it's a mitochondrial toxin. The offending agent is actually MPP plus. So MAOB converts MPTP into MPP plus, and that's what causes the toxicity. Actually, people don't use MPTP very much anymore, except for dyskinesia trials, because you really need a comprehensive lesion to be able to generate levodopa-induced dyskinesias. if you use AAV alpha-synuclein, you can't get a large enough deficit to have a robust and consistent model of dyskinesia. So that's where MPTP is being used most these days.
And I just want to add into that conversation, Jeff, that it's an underappreciated fact that it doesn't matter if it's a human, a mouse, a monkey, a rat. You need to have at least 90% death of nigral dopamine neurons or denervation of the striatum, at least on one side, to be able to get dyskinesia. So yes, we're restricted by needing that kind of a model, or in the rat, the 6-hydroxydopamine.
Yeah, that's really interesting. So what do you think is the future in terms of treatment of patients? Where do you think is this going to lead?
Jeff, do you want to talk about comparison of this therapy?
Well, I'm still waiting for a neuroprotective agent to be positive in clinical trials. I think the real big effect of this treatment that we're doing is the elimination, not the reduction, but the elimination of dyskinesias. I always state that if levodopa didn't have any side effects, that a lot of these stem cells and gene therapy approaches wouldn't be necessary, because you could drive someone to an excellent on, and that would be sufficient. You wouldn't need to have brain surgery. The other thing I think is interesting that's emerging, and I'm doing some work in Madrid with Jose Obeso, is that you could deliver the levodopa non-incisionally. So you can use focused ultrasound to open up the blood-brain barrier of the putamen and intravenously deliver the viral vector and get it into the brain. Now, we're working to optimize these things. But I think that the delivery systems will be changing. And once we can optimize that, I think we'll be able to much better proceed into the clinic with all of these approaches. All of them deserve their day in court, and let's see, what is best. But I can see the first thing we have to demonstrate in humans is safety and tolerability in a phase 1b clinical trial. Then you do an efficacy trial. And although there's now a pattern in which some people do the phase 1b safety and tolerability and go right to a phase 3 clinical trial. And that's been approved by the FDA. And so to get into the clinic in larger numbers, I think will be much easier going forward. But I think and also combinatorial therapies may be best. So if you give the CAV 1.3 shRNA with GDNF, say, maybe that would be an optimal approach going forward.
I see. So what are the trials shown with using neurotropic factors? Has that worked out pretty well or not?
Well, not Chris Bankovich is get the name of his company right now, but Bayer is controlling this. Is in a phase 2 clinical trial, and what they're finding, the phase 1 clinical trial showed a stabilization of UPDRS scores over a number of years. Now, open label trials are fraught with problems. They fraught with a placebo effect and some experimental bias. And so you don't know what effect is real or not. And but there is optimism going forward. And we will have to wait to see this phase 2 clinical trial results that I think will be delivered in the not too distant future.
Nice. That's really interesting. So if you were to go forward with a trial, what additional animal preclinical studies do you need in order to enter a phase 1 trial?
Well, first of all, we need to do safety and tolerability studies. That is the number one. There's no way the FDA is going to let you move forward without demonstrating safety at multiple doses. Now, Kathy has led a grant that Fredric and I are on, in which we are doing that. We're doing safety and tolerability studies at multiple doses. Also, again, delivery is a very key point, and we don't know whether the construct that we're using now is the best. And so we're going to be comparing different constructs. The FDA doesn't care about efficacy all that much. They just want to make sure no one gets hurt. But I think once we finish those studies, we're going to do an efficacy study and go in the clinic as soon as possible after that.
I'll just add in here that, this is a good place to bring up that we recently received an award from a foundation. It's a independent nonprofit organization called Spark NS, and they're dedicated to efficiently educating academic scientists to allow us to advance promising academic discoveries that are found in the field of neuroscience, especially Parkinson's disease, to go from lab to clinic. And so I can't give you any details on this, but I can tell you that they're providing us with milestone-based funding, education in development of our ideas to bring it towards the clinic, mentorship from industry experts, and networking opportunities that, most academic scientists absolutely don't have access to. So we're hoping that all of this education will allow us to talk or do the things that Jeff was talking about, to put it on track to be brought into the patient. So we also have an RO1. I don't know, Fredric, if you want to talk about what we're doing there with, trying to look at the best patient population to increase our understanding of, who we would target and who this therapy would be best for.
Yeah, sure. So one of the questions that's kind of been on our mind is how do you get a first clinical trial going, and what is your indication? Obviously, we're seeing this quote unquote multimodal benefit in our primate study. And what is the population, and how do we translate that into humans? So the next set of funded studies from the NIH is going to younger monkeys and essentially emulating young onset Parkinson's disease. And these patients, I think, as Kathy mentioned, have a more severe and untractable dyskinesia. And this could be an indication that could be a target to get this into patients initially. But then, of course, the question becomes, and I think Kathy alluded to this, we're seeing retrograde transduction and we're seeing neurorestoration. And the question is whether or not we also want to focus in a little bit more on the nigral neurons themselves. And certainly we're going to look at this in every study from now on to see, are we essentially providing disease modification and neuroprotection specifically?
Is this spread of virus along, neuronal tracts? Is that coming from the basal ganglia into the substantia nigra, or is it the other way around?
Yeah, I mean, by any stretch, it has to be retrograde transduction from terminals, right? So we do intraputaminal injections, and whatever terminals are remaining have the ability to take up the virus. And we see this in most animals, regardless of the status of the circuit.
Right. I guess I have one final question, which it seems to be very important, which is the inflammatory response that's associated with the disease. And I wonder what your thoughts are about how important this is, and does your therapy also reduce inflammation?
So inflammation is extremely important. And one of the things I think about some of the trials with immunotherapy trying to clear synuclein, you can clear synuclein, but what about tau? What about TDP-43? You're not clearing all these other misfolded proteins. So you have to get at a place where all the pathology kind of funnels to. And that's, for me, two things. One, lysosomal function to increase the ability to clear them, and also neuroinflammation, because they all have neuroinflammation. And I can tell you the neuroinflammation comes first before the synucleinopathy. I know that from my fetal transplant days, and I've looked at a number of post-mortem cases, and you get inflammation very early. But the Lewy pathology that occurs in fetal transplant doesn't occur for 10 years. So getting at the neuroinflammation early, I think, is a very important therapeutic target.
And we would predict that, with the retrograde transport back to the nigra and, restoration of phenotype, which would indicate that those cells are healthier, that we would be reducing, inflammatory oxidative stress measures. And we have studies ongoing in some of our postmortem tissue that we're going to be looking at that, as well as some future, studies that will more directly look at that. But inflammation has been strongly linked to levodopa-induced dyskinesias, and therapies that can reduce inflammation are, shown to reduce dyskinesia. So I guess by that notion, since, we're preventing 100%. The induction of the levodopa-induced dyskinesias, perhaps that indicates that we are reducing inflammation, but yeah, we don't know anything about that directly ourselves.
Right, I see. So I was going to ask if in the use of your therapy in monkeys, have you seen an impact on inflammatory processes?
Yeah, we haven't really looked at that in much detail. That's, something we have ongoing with this. We have a R01 competitive renewal that we were recently awarded. So that's, some of our future studies for that.
Yeah.
But I think the important aspect of that too is, as what we learned from the human studies, is you're not making inflammation worse by introducing the virus. And that's
Absolutely.
important, obviously.
I see. Yeah. What kind of doses are you using in your animal studies? Pandemic, it's huge, right? So, but it's local, right? So I don't know, quite 10 to the 11th or something like 12 particles.
Yeah, we're using, don't remember the volumes now, but our titer is 1E12 per milliliter. And we're spreading out the injections, so it should be pretty uniform throughout. And certainly this is nothing you would be able to do systemic. There's so many calcium channels throughout the body that, for instance, the
Right.
heart. So this is, I mean, this is the quintessential gene therapy because you have to target this very specific circuit. So,
Yes, and if a drug, which is there's a very severe scientific trying to develop CAV 1.3 antagonist drugs, and if that's achieved, because currently there are no effective CAV 1.3 calcium channel antagonists, which is why it's believed that that's why the steady PD therapy trial failed, because there just wasn't strong enough engagement. But even if they can manage to get a more effective therapy, if you give systemic administration of anything that's going to silence or antagonize CAV channels, like Fredric said, there are CAV channels throughout the body, especially in the cardiovascular system, in the brain, in the hippocampus. If you impact those, you're going to impact learning and memory. They're in the suprachiasmatic nucleus and pancreas. They're all over the place. So, like Fredric said again, direct targeting of the striatum and or nigra is what we're going to need for this therapy.
I see. So I think we ought to probably wrap up. I think that the work that you're doing is so important, and I think it provides hope for patients who who are taking the standard of care. And it seems to me that with dosing regimens in animals and safety studies and that sort of thing, that you should be in not too long poised to test this in patients, and it would be a huge step forward, I think, in the treatment of this terrible disease.
If I could just add one thing, Joe, some trials never enroll a patient, and recruitment for some trials are very difficult. I think we'll have zero problems recruiting patients because so many of them have this disabling dyskinesia, and
that's right.
That'll help us with our path forward.
Yeah. It's a tremendously important unmet medical need. So the best for you guys. I hope things get moving. All right.
If I can just, kind of close with, I have a quote in front of me from a Parkinson patient who recently had brain surgery for a different, indication, but he says, brain surgery may sound daunting, but for me, it's less daunting than the daily struggles of living with Parkinson's.
Yes. Okay. Well, thank you so much. You've been a great group to talk with and shed a lot of light on this important subject, and really appreciate it.
Thank you for the invitation to do this.
Thank you.
Thanks, Joe. Good to see you.